Hi, this is Empress. Today we are discussing about the classes of biomolecules affected in disease. So all classes of biomolecules found in cells are affected. So in structure functions are among the one of the another disease. They can be affected of primary manner like defect in DNA. So secondary manner like structure of functions of amount of other biomolecules. So what are the biomolecules? DNA, RNA, proteins, lipid and polysaccharides, glucose, amino glycans and electrolytes and water. These are the biomolecules. So rate of biomolecular alteration, the biomolecular alteration that causes disease occur rapidly or slowly. The cyanide inhibits the cytochrome oxidase, kills with the like a few minutes, like a massive loss of water electrolytes and cholera can be threatening of life within hours. So it may be take years of build up the biomolecules effect on organ functions. So example, mild cases of Niemann-Pick disease and may slowly accumulate like a sphingomyelin in liver and spleen. Deficiencies of excess of biomolecules. Diseases can be caused to deficiency of excess certain biomolecules. The deficiency of vitamin D result it rickets and excessive result of potential serious hypercalcemia. Nutritional deficiency, primary cause and secondary cause. Primary cause coming through food diet and secondary cause the inadequate absorption, so increased requirements of inadequate utilizations and increased inadequate this. So organelle involved like almost every cell organelle has involved in the genesis of various diseases like nucleus and different mechanisms, similar effects. The different mechanisms like biomolecules can be produced the similar pathological, clinical and laboratory findings. The major pathological procedure can be produced by a number of different stimulus. Those are the fibrosis, fibrosis, liver and cirrhosis, and can be resulted in the chronic intake and electrolytic oxidation. So these are the excessive copper Williams, excessive copper means the Williams disease. So excessive iron means that is a primary hemochromatosis. Deficiency of alpha-1 trypsin. So different biomolecular lesions producing the similar and point when locally concentration compound exceeds solubility in point of excessive formations of decrease or removal of precipitations of calculus. The calculus like calcium oxalates, like magnesium ammonium phosphate and acid like uric acid and cysteine may also renal stores. These are the some of the conditions will form accumulate the different molecules like the regions. So some of the conditions will form of the renal calculus, that is nephrolithosis. This is a condition of calcium oxalates, calcium phosphate and calcium carbonates. These are the substance will be formed like hardening substances forms the renal lithosis conditions. So that some coming to the genetic disease, many diseases are determined genetically. There are three classes like chromosomal disorders and monogenic disorders and multifunctional multifactorial disorders. Coming to the genetical disease, the polygenic denotes the decide with the cause of multi-genetic factor independently environmentally influenced. Somatic disorders is the mutant occurs in the somatic cell in many types of cancers like mitochondrial disorders due to the mutations of mitochondrial genome. So then we're talking about the chromosomal disorder. The chromosomal is excessively lost chromosome deletions and the chromosomes translation, and trisomial is Down syndrome, that means XXX. This is a trisomy. This the triple X will calling of recognized by the analysis of karyotyping chromosomal pattern individually is alteration by the large enough to be easily visualized. Translocations of important activations of oncogene like a phylodiplia, like chromosomes like bacteria and ablated genes. Then next we will talking about the chromosomal disorders. Then we will talking about the monogenic disorders. So involves in the signal mutations and classifications of autodominant and autosomal like sex-linked. Autodominant is the clinically evident of one of the chromosomes affected by the heterozygote and familially hypercholesteremia, and autosomal coming to the autosomal, both chromosomes must be affected like homozygous, and sickle cell anemia is an example for the autosomal recessive disorder. And X-linked mutant present on the X chromosome, whenever female the may be either heterozygous and homozygous for effect of genes. The males are affected from the in the inherited and mutated genes like a degenerative DMD, that is the degenerative muscular dystrophy. Then we're talking about the multifactorial disorders. This interplay number of genes in the environmental factors and patterns. Like pattern is inheritance, inheritance does not come from classical Mendelian genetic principles due to the complexity of genetic and harder to identify the effect of genes. Those less known as about the category of disease like essential hypertension. Inbornness of errors of metabolism, the mutant of structural genes of may be affected with structural and encoded proteins. So if an effect of inborn errors like metabolism may be result of the genetic disorder of which is specific effect of producing the metabolic blocks and that have a pathological consequence. So inborn errors of metabolism, a block have three results, that is decreased formations of product and accumulations of substrate like S behind the block, and increases the formations of metabolic X, Y of the substrate, resulting that it is accumulated. Any one of these three may have pathological effects. So the phenylketonuria, inborn errors, the mutant gene is usually like a phenylalanine hydroxylase. The synthesis of the tyrosine are often the fair skin have increased the plasma levels. Phenylalanine excrete the increase the phenyl pyruvate and metabolites. The structure it is, if such structural gene non-catalyzed proteins affected or mutant can have serious pathological consequences. Hemoglobin S. So genetic linkage studies, that means more distance to genes are from each other, like some chromosomes that greater chance recombinations of occurring between them to identify the disease and causing the genes. Perform the linkage of analysis of distinct fragment length polymorphism or the other markers study the inheritance of disease markers. So this simple sequence of separate like repeats like SR simple sequence repeats are microsatellite small tandem repeat units of 2 to 3 2 6 base pairs are informative polymorphism, then there are restricted fragment length polymorphism. So functional approach, the gene initiated on the basis of biochemical defect, so found to the phenotypic defect and HBS and glucose and valine. The evidence of mutant gene is encoding the beta globin, and candidate like a gene approach to gene is whose functions of if lost the mutant could be explained the nature of the disease. The mutations of rhodopsins are considered like causes of blind due to the retinitis of pigmentation. So for positional cloning, there are no functional information about the gene product and isolated slowly it is chromosomal position, information of linkages of analysis. So cloning is CF like a gene based on two markers that is segregated with the effect of individual positional candidates approaches, and chromosomes are like a subregion of identified by the linkage of studies. The subregion like survived to see the what kind of genetic residue there. So with the human genome is sequenced by becoming a method of choice. So this identify the defect of a gene. So one disease gene identify the still we cannot identify the gene defects. So ethical issues, these are the no ethical issues. Here the ethical issues coming to the one genetic defect is identified, no treatment of options. Maybe will patients what want to know the in person like parental screening and will be identified disease of the actually like a Huntington disease and the normal individual. These are the things of polyglutamies of this. So some of the molecular medicine we're talking about the molecular medicine of these things about these things.